Establishing a reproducible method for calculating the magnitude of multisensory integration effects is significant, as it will aid in the facilitation of future translational research across diverse clinical populations. The main advantage to our technique is that we're able to quantify a robust phenotype of multisensory integration that is subsequently associated with important cognitive and motor outcomes in aging, like balance, falls, gait, and executive functions.
Begin by using stimulus presentation software to program a simple reaction time experiment with 3 experimental conditions: visual alone, somatosensory alone, and simultaneous visual somatosensory. Use a stimulus generator with 3 control boxes. The left and right control boxes contain bilateral blue light emitting diodes that illuminate for visual stimulation, and bilateral motors with 0.8 G vibration amplitude that vibrate for somatosensory stimulation, as well as plastic housing for the stimulators. Next, place a center dummy control box equidistant from the left and right control boxes, and affix a visual target sticker to serve as the fixation point. After the experiment has been set up, escort the participant to the testing room. Have the participant sit upright and comfortably rest their hands upon the left and right control boxes. Strategically place index fingers over the vibratory motors mounted to the back of the control box and thumbs on the front of the control box under the LEDs to not block the light. Ensure that the somatosensory stimuli are inaudible by providing participants with headphones over